More than 1 million uniform Femtoliter droplets are produced on a finger-sized substrate, and single-D molecules are randomly distributed into each droplet. The product yield is proportional to the number of D molecules in a droplet.
Our technique can be used for rapid and quantitative measurement of enzyme activity without complicated protein expression and purification.
To begin, set a cover glass on a staining rack and place the rack in 8 molar sodium hydroxide. Sonicate the cover glass and staining rack for 15 minutes at room temperature. Remove the staining rack from sodium hydroxide and rinse the cover glass with water 10 times. Dry the cover glass with an air gun. Then bake and dry the cover glass on a hot plate at 200 degrees Celsius for 5 minutes. Prepare 0.05% aminosilane solution. Immerse the cover glass in this solution and incubate it for 1 hour at room temperature. Rinse the cover glass 5 times in pure water. Then dry the cover glass using an air gun. Bake it on the hot plate at 80 degrees Celsius for 5 minutes. Place the glass substrate on the customized vacuum chuck for the spin coater. Dispense 70 to 90 microliters of type A Sci-Top polymer at the center of the glass substrate. Then immediately spin coat the polymer.
The initial polymer coating determines the quality of the final microchamber array. Dispense the polymer onto the center of the cover glass without introducing air bubbles.
Bake up the coated glass by holding it at the corner.